Okay, so welcome everybody who has joined so far on this webinar. So this is a webinar entitled Crop War Relative Conservation in Protected Areas in the SADC region. So I would like to first of all welcome everybody to this seminar, which will run over 2 days. So today is the first day, and we'll have at the same time tomorrow the second day seminar. Before we start on the presentation, I'd just like to say that we're going to have 4 presentations and then followed by a Q&A session. Okay, I would like to inform you that for the Q&A session, we will ask the people to raise their hand. There is a button for raising your hand on the GoToWebinar, and otherwise you can give you can type your questions in the chat box. Okay, so we will give priority to those who have raised their hands to speak, and afterwards we will address the various questions that we have. So without further ado, I would like to pass over to our first speaker, who is Christine Mendel from the Bioparma network. Over to you, Christine.
Thank you, Ehsan. Good morning, good afternoon, everybody, and thank you for joining us today. My name is Christine Mensel, and I'm coordinator of the Bioparma program for the Eastern and Southern African region. And you may be wondering a bit why we have a big Bioparma slide up when this is actually on crop wild relatives, but I hope I will explain that a little bit in my few words that I'm going to say now. So it's a great pleasure to be with you here this morning, and also to be collaborating on this exciting topic with the University of Birmingham and the Alliance in Bioversity and CIAT. I would also like to extend a warm welcome to all of you on behalf of IUCN, the IUCN Regional Office for Eastern and Southern Africa, which is my home, so to speak. The collaboration on this event is effectively a result of an event that was hosted by the SADC Secretariat to introduce various projects and programs working in the SADC region with SADC member states. And IUCN, as well as also Bioversity, presented our respective areas of focus and projects. And we then realized that there would be much merit in collaborating as Bioparma in particular, so this is the project that I'm representing, because this has a strong link with protected areas in the region. And obviously, the focus of this webinar is very much on working with protected areas and creating an understanding of crop wild relatives within the protected area audience as well. And so basically, Bioparma has the link to the protected areas, and the Darwin project has the expertise on the crop wild relatives. And they're also mapping distribution of these in the regions with various overlaps with protected areas in particular. And that is also the reason why we are working together on this and presenting this together. So this online event was developed by the Crop Wild Relative SADC network, and in part has been made possible by the Biodiversity and Protected Areas Management Program, Bioparma. And to set the scene a little bit more, I also want to say a few words about what Bioparma is. So Bioparma is an initiative of the Organization of African, Caribbean and Pacific States, and is financed by the European Union's 11th European Development Fund. It is implemented jointly by IUCN and the Joint Research Center of the European Commission, additional partners in each of the regions of the ACP, which is Africa, Caribbean, Pacific, hosting the observatories for biodiversity information. In our region here in Eastern and Southern Africa, this is the Regional Center for the Mapping of Resources for Development, or RCMRD for short. And we're working with them in this region, and they're hosting the Regional Resource Hub, as we call the observatory here. The broad goal of Bioparma is to address priorities of the ACP region for improved management and governance of biodiversity and natural resources, with a focus on 3 main areas of work. And I'll just shift my slide. I hope it shifts. Oh, hang on. Next slide. So the areas of work in Bioparma, the first area of work is the Regional Resource Hub that I've just mentioned, hosted by the RCMRD. And this incorporates what we call the Regional Reference Information System, which is developed by the Joint Research Centre, which provides information and tools to support decision-making for effective protected areas management. This is also where we're looking to integrate the information on the distribution of crop wild relatives in this region. So as I mentioned in the beginning, University of Birmingham is working on mapping this. And so that is something that we wish to support the program with by making that available through the Regional Resource Hub and allowing also that interrogation between where these crop wild relatives exist and where they are in protected areas, in fact, and where therefore there is a need to potentially focus some attention on management of these resources in that protected area. The second area of work in the Bioparma program is capacity building through training as well as knowledge and information exchange. Most of that obviously is currently online, such as this event also. But in the past and hopefully also in the future, we will hopefully also have some more physical training events. The main focus of our training in Bioparma is on two main areas, data management related topics that relate then to the development of the Regional Resource Hub and making important information available to decision makers, as well as then direct topics related to protected area management and governance. So that's where this excuse me, joint webinar falls into. This is really to support protected area management. And as crop wild relatives are critically important for food and economic security, and many of the distributions, as I mentioned, may overlap with protected areas, we felt that it would be really useful to join hands on this and provide some in-depth knowledge to the protected area sector on crop wild relatives and their conservation, and put you in touch, essentially, put these two networks in touch. And the third area of work for Bioparma is what we call the action component, which is a competitive granting mechanism that seeks to improve on-the-ground management and governance in protected areas. And projects are selected based on clearly described priority needs defined through systematic assessments of the area. So again, in terms of crop wild relative conservation, this may be something that could be integrated into priorities for a protected area, and thus also become more of a priority for seeking funding. So in summary, Bioparma seeks to support more effective and fair protected areas through assessments, planning, decision, and action. That's our overall goal. And we see collaborations such as this as extremely important and relevant in achieving these effective and fair protected areas that contribute to a society as a whole. So if you haven't done so already, please do visit the Bioparma website for more information. It's on the bottom of the screen, and in fact, the bottom of the screen here. So feel free to visit and find out a bit more about Bioparma. Okay, so coming to today's topic, besides the few obvious intersection points that I've already mentioned, why do we see such a strong link between protected areas and crop wild? I'll just touch on this very briefly, and the following presenters will obviously delve into this in more detail and reiterate some of these points too. So protected areas obviously contribute significantly to the overall economy in many countries in our region, a lot through tourism in particular, and but also other wildlife-based activities. Crop wild relatives are essentially another piece of the puzzle, so to speak, where protected areas are providing a critical service to society by protecting important resources that feed into the food security for our region and beyond, in fact. So crop wild relative populations are potential sources of traits that may be used in crop improvement, such as resistance to pest or diseases, yield improvement, or tolerance to abiotic stresses such as drought, heat, or flooding. So these are adaptive traits which are particularly important for providing stability and in the changing environment of today. So we have climate change, obviously, as a big factor that is affecting much of our lives, and also obviously affecting our vegetation quite severely. And so some of the traits that exist in crop wild relatives are really important for being able to adapt also our agricultural and food security activities. So it's been shown that in the SADC region, it's a diverse region for wild relatives of a number of crops of regional and global importance, with over 1,900, so 1,900 wild species related to coffee, cucurbits, eggplant, lettuce, millets, okra, pulses, rice, sorghum, watermelon, and many others. Despite their unique value for food and economic security, crop wild relatives are not adequately conserved in situ. So that means in their natural environments. So this is again now where the strong link to protected areas comes. So even if they do occur in existing protected areas, we are often not yet aware of that. And therefore, this initiative and also Bioparma's involvement is really trying to bridge that knowledge gap a little bit and ensure that we are aware of where they exist and that we can then act on that in terms of active management. So we see this as an opportunity not to be missed, not only because it adds significant value to these areas, but also because crop wild relatives can be effectively conserved with minimal additional effort and investment and have such a big impact in the bigger scheme of things of food security. So with those few words, I would like to thank the Bioversity, CIAT and University of Birmingham team for this opportunity to briefly introduce Bioparma and wish you all a productive webinar session. Back to you, Esa.
Thank you very much, Christine, for your presentation on Bioparma. I apologize, I did not show my video earlier on in opening the webinar. So thank you very much, Christine. So now I'm going to give you a quick overview, introduction about the in-situ conservation of crop wild relative, and more particularly about the work that we have been doing in the SADC region on crop wild relative. So can you see my screen? Yes?
Yes, we can see.
Okay, thank you. All right. So first of all, I would like to start with some acknowledgement. I can't get this bar to get out of the site here. Oops. Sorry about that. So I would just like to start with some acknowledgement. First of all, just thank Christine Menzel for the collaboration with the Bioparma network. I would like also to thank all the speakers at these 2-day seminar from the University of Birmingham, Joanna Bram and Nigel Maxted, from the University of Rey Juan Carlos University from Spain, Jose Iriondo, to my colleagues at the Alliance of Bioversity and CIAT, Gloria and Prishni Bisesa. I would also like to extend my thanks to all our country partners who are participating in the Darwin SADC Crop Wild Relative Project, from SPGRC, from the National PGR Centers in Malawi, Tanzania and Zambia, as well as to our steering committee members, Lefu Lebesa from Lesotho, Chike Mba from FAO, Hannes Dempewel from the Crop Trust, Michael Aberton from IITA, and Domitila Raimondo from SANBI and also IUCN. And also thank our IT unit for setting up this webinar. Oops. So as As you're aware, the subject of this talk is about crop war relative, and this talk is really targeted towards the protected area community. So protected area community, the protected area managers and staff, very often they are not aware about the existence of these crop war relative within their protected areas. So it is important that we talk a little bit about that. What are crop war relatives? Well, essentially, crop war relatives are just normal wild species. The only difference is that they are related to our crops, including our wild, not our, the plants, cultivated plants, wild ancestors. And they are, as Christine was saying earlier, they are an extremely important group of plants because nowadays they are really a foundation of our agriculture. They provide genes to resistance genes against to fight pests and diseases. They also have adapted traits that can help to mitigate climate risk, and also having other diversity for adding nutritive value and other important characteristics of our crop plant. Just to give you an example from wheat, the value of crop Y relative of Y relative of wheat has been used over many decades now to transfer important traits of many different diseases from the wild species, which is the Aegilops here, into our cultivated wheat. So I would like to acknowledge the contribution of Nigel Maxted for this slide here. And yet, these crop war relatives, the value that they have contributed to agriculture is tremendous. Some past estimation of the value in dollar has revealed that they contribute to over 115 billion US dollar towards increased crop yields per year to agriculture. So this is really a astronomical figure that crop war relative um, provides to agriculture and ensuring food security in the world. And yet, when we look at the populations of those crop war relatives, a lot of them are threatened in the wild. A recent assessment of crop war relatives in Europe, for example, which was undertaken by University of Birmingham, led by Sheila Kell, has shown that over 16% of the species are threatened or near threatened, and 4% of the crop war relatives in Europe are critically endangered. So, and there is a whole range of different threats element that is causing this threat. And crop war relatives are not very well conserved. Christine talked about that in her opening remarks. When we looked at the accessions of genetic resources that are found in gene banks in ex-situ conservation facilities, crop war relatives only represent about 10% of the total germplasm accessions. And of the globally important crop war relatives, a third of them has no accession whatsoever in gene banks, and a third has less than 10 accession. So there is a high percentage of high priority crop war relative that needs to be collected and conserved in gene bank. When we turn to the diversity in situ, here the projects that are geared to protecting them in situ are virtually non-existent. There are just a handful of projects that are addressing the in situ conservation of crop war relative. And even the crop war relatives that are found in existing area, a lot of them are being conserved passively. They are not being monitored and they are not being actively managed. As I said, there are very few examples of protected areas that have been established in the world, for example, in Costa Rica for wild beans, in Mexico for wild wheat maize, and in Peru for wild potatoes. And there are also other examples in other parts in the world. And even in those genetic reserves, very few of them actually meet the standards at which they need to be managed. Iriondo et al. Has developed certain guidelines for maintaining those wild relative populations, and these are not really being applied. We will hear from Jose Iriondo in this webinar tomorrow, talking about these new guidelines for managing crop wild relatives population. The issue about crop war relative have actually been recognized by many global policy instruments. The recent IPBES report on the global assessment for biodiversity has recognized the catastrophic loss of biodiversity generally, including crop war relative, and stated that the loss of crop war relatives of crops will have really drastic impact on agriculture, and therefore the need to conserve them. The global strategy for plant conservation under the CBD have also given prominence to conservation of crop relative under their target 9. The same also under the strategic plan for the 2011 to 2020 biodiversity strategic plan for the CBD, and in particular ASHI target 13 explicitly referred to wild relatives of crops and also domesticated animals. The International Treaty on Plant Genetic Resources have an Article 5.1d that promotes the in-situ conservation of wild crop relative. Also, the FAO second global plan of action has a priority activity 4 that deals with the promoting in situ conservation and management of crop wild relative in protected area, and have indicators actually to look at progress that countries are doing to conserve their crop wild relatives, and countries need to report again those indicators. And also in the Sustainable Development Goals target 5 also talk about maintaining of genetic diversity of seeds and cultivated plants and farm and domesticated animals and their related wild relative. So as I said here, in FAO fora, a lot of discussion has been happened in the past decades about how do we put in place measures for ensuring the conservations of crop wild relatives. The idea of developing a global network for the conservation of crop wild relatives and also land races has been debated within the Commission on Genetic Resources, and these topics are still being happening, and in a few months' time, there will be a symposium that is being organized by FAO on the in-situ conservation of crop wild relative and on-farm management that will further discuss this aspect. But we also, in parallel to this, been trying to develop certain projects at the national level, but also at the regional level. One of those projects, which was led by Bioversity International with the support from the EU and the ACP group, and in partnership with the University of Birmingham, we developed a proposal on in-situ conservation of crop wild relative in 3 ACP countries in the SADC region. This project took place between 2014 to 2016 and brought together a few countries like Mauritius with the University of Mauritius, Zambia with the participation of ZARI, and South Africa with the participation of DAF and also SANBI to develop strategies for the conservation of crop wild relative. some of the key inputs of those projects has been to develop capacities within the region and broader, not only these three countries, but for the whole SADC region country members on in situ conservation, and also on looking on how we may use the germplasm from those crop relatives in breeding programs in terms of pre-breeding. This was done under this project. The countries has also developed detailed checklist and inventory for crop relative. The checklist and inventory was also done at the regional level, and as Christine was mentioning, we identified through this project more than 1900 crop relatives of priority crops for the region, and identify also sites where they are located with a view ultimately for establishing a network of crop relative sites. As part of the project, we developed this interactive toolkit for conservation on crop relative that has now been published on the crop relative portal. Joanna Brem will talk a little bit more about that. And as I said, each of those countries has been developing exemplar national strategies and action plan for the crop relative in Mauritius, South Africa, and Zambia. So our vision then was to upscale this project and create this regional network of crop relative important sites and stakeholder within this region. And for this, we work very closely with the SADC Secretariat and especially the SADC Plant Genetic Resource Center. And we developed a project under the Darwin Initiative. And this Darwin Initiative project was successful, was funded, and it started in 2019. And in this case, we partnered together, obviously, with the University of Birmingham, with SPGRC and Zambia to ensure continuity, and also since SPGRC is hosted in Zambia. And we brought in two new country partners, namely Malawi and Tanzania. But the main objective, one of the main objectives of this project is really to establish a regional crop relative network of in situ conservation in the Southern African region. And here we've been working very closely with the SADC Secretariat. We have developed already a white paper on the establishment of the network that has now been endorsed by the technical committee of SPGRC and also by the directors committee of the SADC Secretariat. And this white paper is now going to be tabled at the Council of Ministers of Agriculture within the SADC for their approval. And once this is approved, then we will be able to establish this network within the region. The other important objective has been to identify these priority conservation site, at least updating this from what we did in the previous project, and further develop national strategic action plan in other countries. So we started with Malawi and Tanzania, and Zambia came forward to revise their national strategic action plan and republish this again. We've also, in this project, developed linkages with ex-situ conservation by strengthening the SADC plant genetic resource gene bank in Lusaka and linking very closely with other gene banks in the 16 countries. Another important aspect is not only linking in situ to ex situ, but our ultimate objective is really to ensure that these genetic material found within those crop relatives are able to be used by farmers, especially by breeders, for making crop improvement programs. And one of the key stumbling blocks, I may say, is getting access to those material. So within this project, we are also consulting and discussing with the Nagoya Protocol Unit of the CBD and the International Treaty on Plant Genetic Resources to develop this aspect of access and benefit sharing for access to in-situ material, including crop war relative. We've carried out a survey, and Gloria Atieno will be talking about this in her presentation later on today. And also we are trying to organize later on in the year a workshop co-organized with the CBD and the treaty and SPGRC and with the national focal points on those protocols and treaty on the subject on access and benefit sharing. Another aspect that we are looking into is also looking at incentive mechanism for enhancing benefits of farmers for conserving crop war relative within their areas close to their farms. And finally, another objective is to develop gendered capacity within crop war relative on in-situ conservation. And this webinar is really about contributing to this, particularly targeting protected area managers, on what they can do to help conserve these crop war relative population within their protected areas, or even within outside protected areas that are under their control. So looking ahead then, we see that while relatives are really key to sustain crop production for the future, and to do this, it is absolutely essential that we engage with all stakeholders. We need that collaboration between the agricultural sector, environment, forestry sector to be working together, as well as with regional organizations like the SADC Secretariat and SPGRC, and with inputs from international agricultural organizations such as ours, the Alliance of Bioversity and CIAT. So we do need to integrate our approach to conservation and use to ensure that they are conserved in the wild, so as we allow that diversity to co-evolve with changes in the environment and cope with climate change and all that. We need to back them up in gene bank to facilitate their use by breeders. We also need to link crop war relatives populations to breeders and farmers. And ultimately, as I said, in this project, our principal aim is to create this regional and national network of crop war relative for in-situ conservation of crop war relatives. So thank you very much for your attention. So with this, I would like now to give the floor to our first speaker, our first presentation, detailed presentation, which will be given by Joanna Magos Brem from the University of Birmingham. Thank you, and over to you, Joanna.
Hello, just a moment, just to make sure that everything is set up correctly. Can you see the presentation and myself?
Yes, we can see.
Yes, we can.
Okay. Hello, good morning or good afternoon for most of you probably. My name is Joanna Magos Brem and I'm from the University of Birmingham in the UK. And in this presentation, I will talk about crop-wide relative conservation in Southern Africa. So the objectives of this presentation are to give you an overview of the crop-wide relative conservation planning. And I presume most of you are involved in protected area management in some way. And I think it is important to know what conservation planning of crop-wide relatives involves and what is behind the selection of specific areas for active conservation of certain species, of priority species. And the other objective is to present you the main results we obtained in planning for the conservation of crop-wide relatives in the SADC region. And I presume this is the most interesting part. Which species are priority for conservation in the region and where we should conserve them. So I will give you a brief introduction. of conservation planning. I will introduce you the concept of national strategic action plans in this particular context, the steps in corporate relative conservation planning, conservation planning tools that we have developed more or less recently, link conservation to utilization, and finally the establishment of the SADC network for corporate relative conservation. So efficient conservation planning is a critical first step to maintain corporate relative diversity for the future. And this basically means to recommend a network of in-situ conservation areas that conserve priority taxa, but also to complement with ex-situ collections of genetically representative population samples in gene banks. Before we go into the details of conservation planning, I would like to introduce you to the concept of National Strategic Action Plan for the conservation and use of cropped relatives. A moment. So in this image, you see that we start with plant diversity towards conservation planning, where, as said, we finally recommend an in-situ conservation network, as well as populations for ex-situ conservation in gene banks. There are some blanks in this image, but I will fill these as we go along. So the conservation recommendations that result from conservation planning are central to national strategic action plans. And these NSAPs, they define a coordinated, systematic, and integrated approach to in-situ and ex-situ conservation. So in practice, this is the document which will establish which species are priorities for conservation, but also where they should be conserved and how conservation of these species should be resourced and monitored. So national strategic action plans, or NSAPs, aim at raising awareness of the value of national cropped relative diversity. They review the suitability of the current policy environment to support the governance structure and identify gaps and needs for policy change. They also provide a framework and roadmap for long-term conservation and sustainable use of cropped relative diversity, define specific actions and resources required to effectively conserve and use this diversity. They integrate cropped relative diversity conservation into existing national, regional, and global initiatives. And finally, they contribute to regional and global efforts in cropped relative conservation and use. In these slides, you see a model that we have been developing for and refining for many years for cropped relatives, and it shows how we go from conservation planning to the development of a national strategic action plan, to its implementation and utilization. So what I want you to retain from this slide, which is a bit complex, is that the conservation planning includes these major steps here in this circle. So the creation of national cropped relatives checklists, their prioritization for active conservation, the creation of inventory of priority cropped relatives, the analysis of its diversity, which can include identification of hotspots, the analysis of the impacts of climate change in the species distribution, the identification of gaps in the priority species conservation, the impact of climate change in species distribution of cropped relatives, and finally, the establishment of cropped relative conservation goals. So basically, we want to answer specific questions in cropped relative conservation planning. And in this slide, I show you a summary of what questions are and how this can be answered. So first question is, which cropped relatives exist in a specific geographic region? And for this, you generate a cropped relative checklist. Then which cropped relatives are more important to conserve? So you select priority cropped relatives and create an inventory. Where are priority cropped relatives located, and are there any gaps in their conservation? And for that, you can do several different analyses like diversity, gap and climate change analysis to select target sites for conservation. And finally, you do your core recommendations for in-situ and ex-situ conservation, which are basically an answer to the question, where should we actively conserve priority crop relatives in situ, and where should we collect them for ex-situ conservation? So here I briefly show you how we can develop crop relative checklists and prioritize them for active conservation. A crop relative checklist is basically a list of all crop relative taxa that can be found in a specific geographic area, say in a country or a region, like the SADC region. And on the left-hand side of the slide, you can find the various steps generally undertaken to produce this checklist. So you first determine the geographic scope, which can be a country, a region, or even sub-country region. Determine the scope of this list, whether floristic or monographic. So floristic includes all crop relatives that occur in that region, or monographic refers to this first selection of the crop genera, and then find out what crop relatives or wild relatives are related to those crop genera. You also get a flora checklist, and you match it with the target crop genera, and from that you then obtain the crop relative checklist. You then annotate this checklist and for prioritization, and make the checklist available to users. Once the crop relative checklist is ready, we then need to prioritize for conservation. As we all know, human and financial resources are not available, generally limited for conservation. So we need to define which priority species we should conserve first. So first we define the criteria we want to use, and generally for crop relatives we use the socioeconomic value of the related crop. So which crops are most important in that region or in that geographic unit. You use the utilization potential for crop improvement. This is a second criteria. So basically, which crop relatives are most likely to contain or that have been used in crop improvement so which crop relatives are most likely to be used in crop improvement, and that can be based on either whether the fact they have been actually used in crop improvement, or whether there is some research on potential traits that are important for breeding. And there's a third criteria, which is a level of threat, so whether the species is threatened or not. Obviously, there are many more criteria, but this can be, there should be defined right from the beginning which criteria we want to use, which ones we think are most important to use at national level or at that geographic region. So then we define a prioritization method. There are two main methods, the scoring method and the sequential. And the scoring, it's basically assigning scores to the criteria to each species or to each taxon, and then sum all of them and get the final score. And so the top species for conservation could be those ones that are in the top of the list with the highest number, with the highest scores. But there's also a sequential method, and this means applying one criteria after the other. So say, for example, you define what are the most important crops that you want to prioritize, then you extract the crop relatives of those crops, then you get a subset of wild species, of wild relatives, and apply a second criteria, like selecting those with the highest utilization potential for crop improvement, and then you get a subset of that previous set of species, and so on. So this is a sequential method. So basically, after defining the criteria and method, you apply those to the crop relative checklist. So let me show you some results. This was a paper, a research that was carried out in a previous SADC crop relative project. And this particular part of the research was led by Sheila Kell, and this was by Eve Allen, Sheila Kell, and colleagues. And so just to show an example of how prioritization was carried out for crop wild relatives in the SADC region. So the SADC region is a very important area for diversity of wild relatives of coffee, cucurbits, eggplant, lettuce, millets, okra, pulses like cowpea, pigeon pea, and sword bean, but also rice, sorghum, and watermelon. And the region has more than 1,900 crop wild relative species. And we had then to prioritize those species, and we identified those species related to crops important for food and economic security, but also those with the greatest potential for utilization in crop improvement programs. This is a summary of how prioritization was done. So in total, 92 food or beverage crops or crop groups cultivated in the region were identified. Then out of those, 745 species related to 64 of these crops or crop groups occur in the SADC region. Invasive taxa to the region were excluded. Then potential confirmed uses in crop improvement were identified for these species and used as a criteria, a second criterion. And we finally identified 100 crop wild relative species, or 113 taxa. That were the final priority crop wild relatives of the region. And the coffee, as you see in this graph, the coffee wild relatives are followed by the cowpeas and millets, are the crop gene pools with the highest number of wild relatives in the region. This is regarding the priority list, of course. But I must highlight that the SADC region contains plant genetic resources which are not only important for food economic security within the region, but also in other regions, such as millet, rice, sorghum, etc. So in turn, the SADC region is also highly dependent for plant genetic resources from other parts of the world, including wild relatives of beans, cassava, maize, soybean, and wheat. But these are some images of wild relatives. So on the left, you have a coffee wild relative taken in Mauritius. This is Coffea Mauritania. In the middle, you have a sorghum wild species that was captured at the edge of a crop field in Zambia. And on the right, you have an eggplant wild relative, Solanum panduriforme, taken in South Africa. So after developing a crop wild relative checklist and prioritized crop wild relative for conservation, we then collate occurrence data for each priority taxon to know where they occur. And with this data, we then undertook several diversity analyses, including hotspots, complementarity, ecogeographic analysis, gaps in conservation and in situ and ex situ conservation, climate change analysis, and finally, we made our conservation recommendations for in situ and ex situ conservation. In this particular slide, you can see the number of gene successions here on your left, and herbarium specimens on the right for the SADC priority crop wild relatives. And the number of herbarium specimens is particularly higher in Eastern South Africa, here, and in Tanzania as well, and as well as in other areas in the region. And here you can see the priority crop wild relatives hotspots based on known occurrences on the left, and on predicted distributions, that is species distribution models here on the right. And both maps show hotspots in South Africa, Eswatini, Tanzania, Mozambique, Zimbabwe, Malawi, and Madagascar. Looking at the complementarity analysis, which is an iterative process that identifies the minimum number of sites that are needed to conserve all priority taxa. We did this kind of analysis, and we did based on a grid or an existing network of protected areas. to assess the representativeness of taxa in the network as well. And this type of analysis can be undertaken at species level or at ecogeographic level. But I will show you some results. Here, some results of complementarity analysis in Zambia by Dixon, Nguni, and colleagues in a couple of years ago. And you see that on the left, they identified 13 grids of 15 by 15 kilometers that are needed to conserve 21 priority crop pod relatives. And when they did the analysis based on the existing protected areas, so selecting the minimum number of protected areas needed to conserve priority crop pod relatives, they identified 10 protected areas that conserve 18. So these ones in green, 18 crop pod relatives, but 3 priority crop pod relatives do not occur inside protected areas, and so 3 additional they needed to identify additional sites for these priority crop pod relatives. So gap analysis, the other type of analysis that we can do for conservation planning, it's a conservation evaluation technique that identifies gaps in conservation, and it basically involves a comparison between the range of natural diversity and the diversity that is effectively conserved in situ. And this is generally called in-situ gap analysis, or with the accessions of the target species that are conserved in gene banks, and this is the ex-situ gap analysis. And again, it can be undertaken at various levels. We can look at the species diversity, at the gaps at species diversity, but also at the gaps at the genetic or ecogeographic diversity level. But I will talk about this a bit later. So for in-situ gap analysis, you first identify those crop pod relatives that do not occur within existing protected areas. And so the gaps are those species that do not occur within these areas. You can also identify species that do occur within the existing protected areas, here in green, but are not actively managed. So these are also gaps because the species might be there, but if it's not managed, monitored, it can vanish from that site without us being aware of that. So this is a second type of gap. And then you can look here in blue, the gaps at infraspecific level. And these are basically the diversity within each species that is not being conserved actively. And for ex situ gap analysis, we identify taxa that are not conserved ex situ, and the gaps are those species not adequately conserved ex situ, but also here in brown, the gaps, we identify the gaps at infraspecific level. So we might have a lot of populations of one species conserved in situ, but they might be just populations that do not cover the broad range of genetic diversity or ecogeographic diversity of that species. So this would be, so those genotypes that are not conserved ex situ would also be gaps in conservation. For in-situ gap analysis, you overlay species distribution or ecogeographic diversity, genetic diversity with a network of protected areas, but you also review in-situ conservation projects to identify which species are actively being managed in situ. And for ex-situ gap analysis, you simply make a comparison between species distribution and the existing ex-situ accessions, but you can also do some ecogeographic diversity analysis, and CapFitoGen is one tool that can be used for that purpose. It's freely available, and you can work this out with this tool. Some results. So for the SADC region, the gap analysis showed that the SADC crop pod relatives are fully conserved, both ex situ and in situ. So 50% of the priority species are not conserved ex situ, and of those conserved ex situ, 40% have less than 5 populations, and 16% have only 1 population in a gene bank. And 17% of the species are outside protected areas exclusively, so they are not known to be occurring within protected areas. And those that occur in protected areas are not monitored or actively managed. Okay, so as I briefly mentioned earlier, as our aim is to conserve diversity within species, not just a lot. We don't want to conserve a lot of species, but we also want to conserve the diversity within species, because this is the diversity that is important for breeders. This is what confers the ability of the species to adapt to different environments. So this is what the breeders are looking for. So we've produced an ecogeographic land characterization map. You can see here on the right, which basically describes the different environments in the region. And here we will have to assume that ecogeographic diversity is a proxy of genetic diversity. So that is to say that different environments mean that the species is genetically adapted to them. And this is when we don't have the genetic information of the species, we can simply take a proxy and use this proxy that is ecogeographic diversity. Of course, this is debatable, but this is the best we can without the genetic information, at least for now. So we used 16 generalist variables and the Kalicki method. The variables are here in the table. And again, we use the CapFitoGen tools to produce this map. So we then looked at how climate change is expected to affect the distribution of these priority crop-out relatives. And in these two maps, we have the predicted change in crop-out relative richness in two different scenarios. On the left, you have a temperature rise relative to the 60s and 90s of 2.06 degrees Celsius, and on the right, an increase of 2.55 degrees Celsius. Yellow to red colors refer to an increase in the number of species lost, whereas the colors green to blue refer to an increase in the number of species. So you can see that there is a slight, well, there is an increase of species lost from one scenario to the other scenario. But basically, in both scenarios, we see that the species loss is predicted to happen really in Southern Africa. And if you look at this graph, you can see the increase and decrease in the number of priority crop-out relatives in each country. So Seychelles, Zimbabwe, and Mauritius seem to be more affected by climate change, with a higher decrease in crop-out relative diversity, whereas Lesotho here in the bottom, Tanzania and South Africa seem to be increasing the number of priority crop-out relatives with climate change. Then if we look at each priority crop-out relative, we have the winners, and those are the ones that are predicted to gain distribution area, but also the losers, and those are those species that are predicted to lose more area than gain. And it is more or less even. And Vigna, for example, Vigna keraudreni here on the bottom, it's very little, but maybe you can see it, a wild relative of cowpea, seems to be the greatest loser, whereas Coffea congiensis there seems to be the greatest winner. This is a wild relative of coffee. And there are taxa that are not predicted to be greatly affected by climate change. This is in the middle here. So Sclerocardia birri and Marula tree, for example. So we have looked at the impact of climate change in species distribution, but we also looked at the different environments in which each species occurs. So how do we then use this information to establish in-situ conservation priorities? So basically, we want to conserve in-situ those populations that are most likely to persist in the future. But of course, looking at the whole range of ecogeographic diversity, if possible. So like in this example, we want to conserve, on the left, you can see ecogeographic land characterization with the Horizon longistaminata, so wild relative of rice. on top of it. And we want to conserve populations that come from diverse environments, like in this first map, as indicated by the arrows. But at the same time, and here on the right, you have a map, a species distribution map of, again, the wild relative Oryza longistaminata, here with the blue dots, and behind that you have the climate change effect, the predicted climate change effect on the species distribution. So in green is light green, the potential new areas for these species, in dark green the low impact areas, and then in red and orange the negatively impacted areas. And so what we want is to conserve those populations that are likely to persist in the future, those ones that occur in the green area, dark green areas. So we then run a complementarity analysis to identify the minimum number of sites needed to conserve all crop wild relatives, and ideally all their ecogeographic diversity, but only populations that are likely to persist in the future with climate change. So our results show that we need 120 protected areas in 13 countries that cover 88 crop wild relatives and more or less half of the ecogeographic diversity. And the most important area, basically the three top areas occur in South Africa. The first top area is the Kruger-2 Canyons National Biosphere Reserve. Then we have this second one, very small private nature reserve. I don't know how to say it, but I think it's the Onderstepoort, also in South Africa, and Kruger National Park in South Africa as well. But then we have the Lake Manyara National Park in Tanzania, the Okavango Delta System in Botswana, the East Usambara Biosphere Reserve in Tanzania again. Here it's 6. Then we have plants in South Africa there, the Ruaha National Park in Tanzania, here in yellow, and the Niika National Park in Malawi there, and the Tsizab Communal Conservancy in Namibia. So what about the remaining crop wild relatives and the diversity that is not conserved in these protected areas? So we run a complementarity analysis for those populations, and we basically 151 sites were identified in 11 countries to cover the remaining 21 crop wild relatives and remaining ecogeographic diversity of the other crop wild relatives that is not conserved within, do not occur within protected areas. And the top sites are here, these 3 in the yellow, in the red arrows. The first one in Angola, the second one here, here in South Africa, and the third one in Tanzania there. So overall, Tanzania, Democratic Republic of the Congo, South Africa, and Madagascar, they probably hold the most important areas for conserving crop wild relative diversity predicted not to be negatively impacted by climate change. So as for ex-situ conservation, we divided it into 2 priorities. So first priority correspond to areas areas of ecogeographic diversity not conserved ex-situ, but that is likely to disappear with climate change. And we see here in this map, there are not many areas in that situation, but here it's probably, if we do the close-up here on the right, we see that this area in DRC and touching Angola there are probably the most important areas for conserving this ecogeographic diversity predicted to get extinct with climate change. But we also observe that 39% of crop wild relatives are likely to lose ecogeographic diversity with climate change. And the millets, the cowpeas, and the eggplant wild relatives are the most affected taxa. We then have priority 2, and those are those crop-out relative predicted richness areas of the remaining ecogeographic diversity, not conserved ex situ, so not unique to areas negatively impacted by climate change, but all of the diversity that is not conserved ex situ. And in this image on the left, you see that the ex situ conservation gaps or the are mainly located here in Angola, in DRC, in Tanzania, South Africa, Madagascar, etc. And as for the remaining diversity that is not, so basically the ecogeographic diversity gaps for the remaining 35 taxa that were not included in the analysis. Maybe I forgot to say that for the species distribution models, we only managed to model 75 taxa out of the 110. Because the other species didn't produce stable models, or did not have enough occurrences in order to create these models. And so on the right here, you see the ecogeographic gaps for the remaining 35 taxa. And for this, we ran a complementarity analysis to identify sites where ex situ collection could be prioritized for this diversity. And you see that these sites there in South Africa and in Tanzania are the highest priority sites for this remaining diversity. So basically Angola, DRC, Eswatini, Madagascar, Malawi, Mauritius, Mozambique, South Africa, Tanzania, Zimbabwe, etc. They are important areas for conserving crop-out relative ecogeographic diversity, not conserved ex situ. So there is still a lot of work to do in the region. Okay, now moving to crop-out relative conservation planning tools. We have developed some tools because they are very helpful to guide and facilitate systematic conservation planning and the development of the NSAPs, National Strategic Action Plans for Conservation Use of Crop-out Relatives. They promote data standardization of data management and minimize errors and time costs. So if I show you this slide again, but now including where the tools were developed, can be used, you can see here for conservation planning, we have developed these three tools: interactive toolkit for conservation planning, a crop-out relative checklist and inventory template, but also an occurrence data collection template, and templates for the preparation of an NSAPs here. They're all freely available online. That can be used for crop-out relatives, so basically to any wild plant group. So if I show you this slide now, the moment. Again, I will show you where these tools fit. So the interactive toolkit goes across all of the steps because, well, we'll show you later. The crop-out relative checklist inventory template helps you generate a plant checklist, but also helps you select priority taxa and produce the inventory. Then the occurrence data collation template will help you with the diversity gap and climate change analysis because it allows you to collect data, population data, and that can be used for this analysis. And finally, National Strategic Action Plan templates, but also a technical background document that goes with the NSAP are here across because they will help you compile all this information. And finally, so the interactive toolkit for crop-out relative conservation can be found in this link. And it will help you go through each and every step in conservation planning. And it includes a brief introduction to each conservation planning step, methodology, which outlines the different components of each step, and also includes an interactive flowchart, which will help you move through the various components and choose the options that are most appropriate, giving your context. And also this toolkit will also show you a lot of case studies for each and every step, references, but also additional resources like tools, software, presentations, etc. It's freely available, as I said. You just need to register, but it's you can use it freely. So to help developing national, even regional crop-out relative checklists and inventories, a series of standards and descriptors have been developed in the projects, were compiled and refined. And these descriptors were then used to develop a tool in Excel. Here, it allows you to create a new checklist, enter data to develop an inventory, etc. It's an Excel because it's more user-friendly for most people. And this is how it looks like. I won't go into the details. It's available. You can download it, and it has been used in these countries. Then the occurrence data collation template helps you to collate and prepare the occurrence data for the diversity gap and climate change analysis. And it's basically it was compiled from existing descriptors. And it allows you to compile occurrence data from various sources. So from herbarium gene banks, bibliography, existing databases, fieldwork, etc. This is how it looks like. It has
Sorry,
been
Joanna,
used in.
can you hurry up a little bit because of the time? Thank you.
Yes. Yeah, almost finishing. It has also been used, and it helps prepare you the data for CapFitogen. And these are the two templates that have been developed to guide you writing the NSAP. One is the preparation of the NSAP itself, and the other one helps compiling all the technical and detailed data that resulted for conservation planning analysis, and that generally doesn't have place in the NSAP. These have also been already used in Mauritius, South Africa, and Zambia. And in Zambia, it has already produced a second iteration for the period of 2020 and 2026. So now completing this slide with the various tools. So we start by plant diversity, conservation planning, where the conservation planning tools have been applied, produce the NSAPs, and finally we conserve the resources and utilize this diversity. So in these last two slides are about the integration of national and regional priorities to create the SADC crop-out relative network. And you can see on the left the priority sites for conservation in Zambia, and the right the national sites from South Africa, but how, and in the middle the SADC sites, but how do we then integrate all these? So the approach we suggested was used in Europe, but also in the SADC region, and considers two different geographic levels and integration of these two levels. So at national level, the country identifies national priority crop-out relatives and sites that conserve these national priorities, and this is illustrated in the green part of this complex scheme. It's important to say maybe that some countries in Africa have already identified national sites, for example, Malawi, Mauritius, South Africa, and Zambia, but several others are currently doing it. And this is the case of Angola, DRC, Eswatini, Lesotho, Madagascar, Tanzania, and Seychelles. Well, these national priorities might not be important at regional level. So in the case of the SADC region, we have already identified crop-out relatives and sites to conserve regionally important diversity. And this is shown in the blue part of this scheme. Obviously, each country will decide whether to establish the important regional sites for crop-out relative conservation. So what we finally suggest is this orange bit. It's an integrated crop-out relative conservation strategy for the region that is a mix of sites of national importance that conserve national priority crop-out relatives, but also sites of regional importance that conserve regionally important crop-out relatives. And this is illustrated here. Okay, I think this is it for now. Sorry for taking long, and thank you.
Thank you very much, Joanna, for this brilliant talk. I would really ask anybody would like to have any clarification to note their question down and hold it until after the next presenter has given her presentation under the Q&A session. So may I now invite Gloria Otieno and Prishni Bisesau, who is going to give a presentation on the access and benefit sharing of in-situ genetic resources within protected area. Over to you, Gloria.
Thank you very much, Eshan, and thank you very much, everybody, for attending this seminar. My name is Gloria Otieno. I'm from the Alliance of Bioversity International and CIAT. I'm a policy specialist, and I'm based in Uganda office. So I hope you are able to see my screen and hear me well.
Yes, we do, Gloria. Karen.
Okay. So can you see the slideshow?
Yes, we can.
Okay. So this work is done in collaboration with Prishni Bisesau, with whom we have been conducting a background study, which also involves a survey. And so I'll just go right into the nitty-gritty of access and benefit sharing from international perspectives. And then I will talk about the work under the SADC on access and benefit sharing of crop wild relatives, specifically within this project. So access and benefit sharing involves a set of rules and principles governing the use and equitable sharing of benefits arising from genetic resources. So at the international level, there are two main regimes of access and benefit sharing. One is through the International Treaty for Plant Genetic Resources for Food and Agriculture, and this is usually for genetic resources in this list of 64 crops governed by the treaty, but also genetic resources in the public domain, which are accessible through standard material transfer agreements. And the other regime is mainly through the Convention of Biological Diversity and the Nagoya Protocol. And so anything else which is not accessible through the plant treaty is accessible through the Nagoya Protocol. And this is mainly for anything else that is not in the public domain. Genetic resources that are in situ or ex situ, but are held by privately or held by communities, and therefore not in the public domain. So I will not delve too much into crop wild relatives, but I would just like to stress the fact that they are very, very important for the traits that we need for breeding, especially with respect to climate change that is happening now everywhere in the world. So they hold important traits for drought tolerance, pests and diseases, and this can be crossed to produce new varieties that are more resilient. And crop wild relatives are found in the wild, and therefore they are very vulnerable to land use patterns, so clearing of forested areas, and also they can be issues of climate change, as Joanna has already talked about, and of course they are susceptible to loss through these man-made or climate change issues. And therefore we need to conserve them, and we also need to use them pre-breeding and breeding. So that is why their access is very important, and that is why the benefits accrued from their access is also very important, because these benefits can be used then to help with the conservation of the crop wild relatives. So crop wild relatives are found in situ in protected areas. And when they are in situ in protected areas, which are governed by national governments, then they are in the public domain. And if they're in the public domain, they can be accessed through the plant treaty, through standard material transfer agreements. But crop wild relatives are also found in private rangelands. That means they are private owners of rangelands, and these are mainly common in the southern Africa. We know many people own private rangelands in Zimbabwe, in Zambia, in Kenya, and in other countries as well. And they are also found in farming communities or forested areas that are managed by communities, and therefore their access depends on negotiations with these owners of private rangelands or with communities, and this is through the Nagoya Protocol. And then crop wild relatives can also be found in gene banks, and that is ex situ. So they are conserved by national gene banks, and that means they are in the public domain. There are also CIAT gene banks that have collections of crop wild relatives, and this is also in the public domain. So these are accessible through the plant treaty. But we also have private sector collections, especially from some big private sector seed companies that have some collections of crop wild relatives that they use for breeding. So those are again accessible through negotiations with the relevant persons. So because of the way the crop wild relatives are found in situ, which means both in the public domain and not in the public domain, the scope of access and benefit sharing is at the international level is mainly through the treaty and the Nagoya Protocol. So under the treaty, we have Article 10 of the treaty, which states that these materials, if they are in the treaty and they're in the list of 64 crops and they're in the public domain, then you can access them directly through the SMTAs or through the national governments. But this is again subject to national legislation. And then there is also the issue of accessing through the Nagoya Protocol, what is not in the public domain. And this is where we have now to negotiate for access with the holders of the genetic resources in situ or ex situ. And we have to negotiate by having prior informed consent. For example, if it is forested areas that are managed by communities, you have to negotiate with them prior informed consent under mutually agreed terms, so that you also agree on the benefits that are accruing to these communities to help them continue with the work of managing and conserving these crop wild relatives. So when you go to the country level and you want to access the crop wild relatives, you first of all have to ask yourself, who has the authority to regulate the access? Is it in the public domain? Is it in the private domain? Is it with communities? And is it found in protected areas that are publicly managed, and therefore you can access it through the SMTA? So then you fall into issues of access permits, and you ask yourself, who gives access to these materials? Which competent authority are you going to send your request to? Because requests are sent to competent authorities, either of the Nagoya Protocol or of the international treaty. And what are the conditions of accessing this? If it is in the SMTA, then it is standard. But if it is you need to negotiate, then you have to agree on the terms and conditions for accessing this material. And then, of course, there are different regulations and different policies, as we will see as the presentation moves on. So you fall into issues of bureaucracies, you fall into issues of who is giving permission and who are the users and who are the providers of these materials, and how they are negotiating and how they are finally accessing the material for breeding or for whatever reason they may want to use them. And so under the CBD and Nagoya Protocol, access is mainly, and I've already talked about this, you have to get permission from the provider and get prior informed consent. And you also have to negotiate an agreement to share benefits resulting from the genetic resources. This is known as mutually agreed terms. And then there is the issue of compliance. You must comply with the national ABS legislation under mutually agreed terms. And then, of course, there are checklists, like you have to comply with the clearinghouse mechanism issues. You have to have checkpoints, and you have to have internationally recognized certificate of compliance. And then, of course, this protocol also addresses the issues of access to traditional knowledge for those material that is in situ and is conserved and managed by communities. Then there is also the issues of traditional knowledge associated with the genetic resources, and also the issue of benefit sharing from the use not only of the resource, but the use of the knowledge, and of course, compliance with national legislation. So this activity under the SADC countries, 16 SADC countries, I'm going to now talk about the objectives of this activity. This specific activity was mainly to conduct a review of policies and regulations in the 16 countries, to identify best practices, but also to identify the existing constraints that exist in policies, in laws, and in regulations. And these constraints are constraints to access and constraints to benefit sharing. So that is ongoing at the moment. And then we have also been conducting a survey with key stakeholders, mainly to identify, again, constraints and gaps. And these stakeholders are mainly users and providers of genetic material, but they are also policymakers in the specific countries. So we have contacted focal persons of both IT and Nagoya. We have also contacted managers of protected areas, policymakers, both in the Ministry of Agriculture and Environment, and in any other ministry where issues of access and benefit sharing fall. We have also targeted breeders in our survey, and also other users and providers of crop wild relatives. Like, for example, if we have very good examples with communities providing in situ materials of crop wild relatives. And then the other objective was, after having these two main issues, that is the background study and the survey, we combine this information to make policy recommendations towards harmonization of access and benefit sharing in the region, in the SADC region. Now that there is a network of crop wild relatives conservation, perhaps we can also have a harmonized way of accessing resources of this nature within the region. And then the other thing we are going to do is hold a workshop to disseminate the findings of these two, the survey and the background study. And maybe during the workshop, propose common guidelines for access and benefit sharing in the SADC region. And these common guidelines, we are going to work together also with the Treaty Secretariat and the Secretariat of the CBD and Nagoya Protocol to find ways in which we can harmonize the principles of access and benefit sharing in the SADC region, because different countries have different approaches, different policies, and different laws and regulations for access, but also different institutional issues that come up. For example, some access and benefit sharing issues fall under Ministry of Agriculture, and some of it falls under Ministry of Environment or with managers of protected areas, but these people, and they don't talk, they are not in any network, they don't associate with each other in any way. So you might find that when you are a user and you are seeking access of these genetic resources, it may be hard and there might be many bureaucracies that hinder the proper access of genetic resources, but also on the benefit sharing, so that it reaches the goal of conservation, and also it benefits the communities that are holding these resources. So some highlights from a review of policies. So we found that only 2 countries are not party to the treaty, the international treaty. So that is South Africa and Botswana. So if you want to access resources related to food and agriculture, let's say crop wild relatives that are related to food and agriculture, you have to use the regulations that they have set up, and that is the access and benefit sharing regulations. South Africa has a separate regulation on access and benefit sharing, and that was the BABS, the bioprospecting access and benefit sharing regulations, which were of 2015, and these were done before they even became contracting parties of the Nagoya Protocol. And we also found that all countries, the 16 SADC countries, are party to the CBD and Nagoya Protocol. So if you can't access the genetic resources in Botswana and South Africa through the SMTA, then you need to access it through the CBD and Nagoya Protocol, which means negotiating with the relevant parties to provide access. And then we tried to look at what kind of ABS regulations are there. So we found that only 2 countries have separate ABS regulations, and this is Namibia and South Africa. And the Namibian one is also associated with the traditional knowledge. So the rest of the countries, the regulation is spread through the Ministry of Environment, the National Environment Management Acts in the various countries, or through the Ministry of Agriculture, if the material is to be used for food and agriculture. And some of it is also found in legislation on the management of protected areas, specifically saying how to access resources in protected areas. So it is not very clear, you can't find really a clear ABS law for in-situ material, but they are scattered here and there. And also the institutions, you find that the competent authority for accessing material in protected areas is under the national environment. But again, if this material is a crop wild relative, you need to access it through the Ministry of Agriculture. So the coordinating issues between Ministry of Agriculture and Ministry of Environment are also flagged out in some of the countries' regulations and laws pertaining to access and benefit sharing. So I've already talked about ABS regulations being found in different acts and legislations. And although one positive thing is that some of those legislations stipulate that anything being accessed for purposes of food and agriculture should be through the international treaty and the Ministry of Agriculture, and using SMTAs, regardless of whether it is held by communities or it is in publicly managed protected areas. And a very good example of such kind of a legislation is the Ugandan legislation of on access and benefit sharing, I think 2007, where anything that is being used for food and agriculture, if you want, for example, crop wild relatives of millet, you are supposed to go through the focal person of the international treaty. and they will grant access using SMTA. So these are some of the best practices we are going to also learn from. And then I will just present something on preliminary results from the survey. This analysis is still ongoing. Prishni and I are still working on the data, and some data is also still coming in. So we conducted the survey in 27 countries of the SADC and Eastern Africa region. So countries like Rwanda, Burundi, Kenya, Ethiopia, Uganda were also included in the survey. And we got responses from 21 countries, and those are highlighted, which responded to the survey. And we had a total of 40 participants responding to the survey, which is a very good thing. And that is the distribution of the respondents. And we had 42.5% of respondents are women, and the rest are male. And then focal persons of the treaty, we got 9 responses. Focal persons of Nagoya Protocol, 5. We got only 1 breeder. Competent authority of the treaty, 2 responses from Nagoya. 5 gene bank managers were able to respond, 3 policy makers, and 3 people who are conserving material. But we got 0 responses from managers of protected areas. And this is a very important constituency for us because they are providers of genetic materials being conserved in situ, and especially the crop wild relatives. So we would like to know what kind of constraints and gaps they face in providing genetic resources. So we would urge the managers of protected areas to kindly fill out the surveys so that we are able to have more insights into the issues of providing genetic resources for breeders and for conservationists. Then we asked the respondents what, just to gauge if they have very familiar or they have never heard of different international policy instruments, and they've pretty much heard about the CBD, Nagoya Protocol, and the international treaty. But most of them, don't have much information on the International Plant Exchange Network, where crop wild relatives are also exchanged. And most of them also don't have or have never heard of principles on ABS under ARIPO, because ARIPO states also have the ABS issues and the common policy guidelines. So those are some of the results we had. And we asked the providers, so how does your institution provide access to crop wild relatives? Most of them are provided in situ under standard material transfer agreements because they are in the public domain, and also because they are related to food and agriculture. And then ex situ under standard material transfer agreements, 25%, and under mutually agreed terms, negotiations also. But we also have those who have materials both in situ and ex situ. And this is mainly the gene bank managers that have herbariums as well. And they also provide, materials through mainly SMTA, but also through negotiations with the PIC and MAT. So what is the category of third parties do they supply in situ material to and crop wild relatives? So we have breeding programs, CG centers, international organizations, and local and international universities for research purposes. and also pharmaceutical companies do request materials from these providers of genetic materials. So we are still analyzing the data, and some of the data is still also coming in. We hope that we will be able to get more insights into the constraints and gaps in accessing crop wild relatives, especially from the providers, managers of crop wild protected areas where crop wild relatives are conserved, but also from the users of these genetic resources. So we intend to also target more breeders and to provide some insights on how they access these crop wild relatives, both in situ and ex situ. But we also hope to identify constraints and gaps in legislation, and also to flag out the best practices, and also to bring out issues of institutional coordination. The questionnaire also asked some questions on institutional coordination. For example, which institutions do you collaborate with in formulation of these policies, or which institutions do you collaborate with when providing access to genetic material, or when negotiating for mutually agreed terms. So those issues will come out as we move on with this analysis of the data. And then we hope that we can make recommendations on what we can do to help both users and providers to have an easy time in accessing genetic resources, but also to see how benefits can be accrued and can be used by national governments for conservation, and also by communities, because communities also contribute a lot to the conservation of forested areas, and how can these benefit sharing options help the communities to conserve, maybe some capacity building, maybe some incentives for communities that they can conserve this and manage these resources well. Maybe knowledge transfer, various types of incentives that can be used at the national level to really help us to conserve these crop wild relatives, because they are very important for our future. And then we will also propose harmonization at the SADC level, and also guidelines for, for common guidelines at the SADC level for accessing and the genetic resources, and also for possibly also for benefit sharing. And for these guidelines and harmonization processes, we are going to engage the treaty and the CBD Nagoya Protocol folks. And I hope now also Bioparma to provide more insights into what we can do to have good mechanisms for access and benefit sharing. Thank you very much.
Thank you very much, Gloria. I didn't want to cut you off. It's a very important presentation about access to in situ genetic material, in particular crop wild relatives. So we've got basically about 15 minutes left for question and answers. I think there has been quite a number of questions being asked in the question box. First, I would just have a look at any raised hand. I see one person having a raised hand, so I will unmute Suzanne Ango for her comments. Suzanne, you have the floor. Valerio, could you unmute Suzanne, please? Suzanne, I think you are self-muted. Please unmute yourself and you can take the floor. No?
Suzanne, I have unmuted you. You need only to unmute yourself. You need to click on the red microphone icon.
Okay, perhaps in the meantime, we can take one question from the question list. Prishni, can you read out the first question?
Yes, so it's a question from Sheila Kell about a presentation from Joanna. So will you be looking at the marriage between priority sites at regional level and national level in this project? That's the first part. And the second part is, will you be investigating other non-priority crop-relative taxa which occur in the same region?
So, Joanna, would you like to respond to this?
So yes, I still haven't looked at the matching of national and regional sites, but it's something that it's important to look at. I know some of the sites. Well, I know because I've been talking to various people in various countries that some of the sites have been already identified nationally. So some of the regional sites have already been identified nationally as priorities. But this is something that we will look into that, because it is an add-on, I think, if a site is both nationally and regionally important. And regarding the other species in the region that might be of importance, we are not thinking of looking into those, but it's probably it could be an extra to this analysis and to this project.
Thank you, Joanna. Maybe I can add something to this. I think in the project, in the Darwin project, we've also been doing a capacity building, a training program, where all these steps that Joanna has just explained in her presentation has been done. And there is ongoing mentorship with individuals within different countries to apply those. So if there are certain analyses that can be done at national level, I mean, that they are welcome to do that.
Yeah, as I said,
Susan,
just to add, as I said in the presentation, there are various countries that, like Lesotho and Eswatini, Madagascar, and so on. They have been identifying these priority species, and they are currently collecting data for then identify national sites to conserve them.
Thank you. So we don't seem to be able to hear from Susan. So I'll give the floor to Sheila, who has raised her hand. Sheila Kell.
Thank you, Aishan. Yeah, it was just to clarify for Joanna that the second part of my question was actually, which would you be looking at non-priority species conservation in the same sites that are identified, so the same protected areas that are identified as a priority for conserving the priority species?
Sure.
Yeah, well, that is a very important issue, because besides those species that we know that occur there might be other species of equally crop-out relatives also occur on those sites. So we are not looking into those now. That would imply to collate occurrence data, but since the protected area managers that hold crop-out relative of regional importance will know which species we looked at, we might give some guidance as well on other crop-out relatives of other important crop genera that might also occur in those areas. So if they probably will know which species they have in the areas, and we can inform them about crop genera. And so they can identify which additional species might be there that might be worth conserving within those areas. So in that sense.
Thank you, Joanna. I have another raised hand from Nigel Maxted. A quick one, Nigel, please. Thank you.
Can you hear me, Aishan?
Yes, we can.
Okay, so I have a question for Gloria. So Gloria, if we're looking at in situ conservation in the region, then it's important just as with ex situ, that in situ is sustainable. And my view is the only way we can actually make sustainable in situ conservation is through making sure that breeders and other users have access to the material in the in situ area. So so your talk was very important. But when we look at the similar situation in Europe, we found that the idea of having protected area managers or individual farmers, if we're looking at in situ or on-farm conservation of land races, them providing the material themselves is unsustainable. I mean, basically, they don't have a history of providing material from their in situ sites. They don't have any training, and basically they're very busy with all the activities they're already doing. So really the way we found round this is to suggest that, if you like, that the gene banks, or we would rather call them genetic resource centers, act as a mediator in provision of in situ material. So I don't know if you've thought about this in terms of the SADC region or how this might be applied, but this way it takes away the responsibility of the local protected area manager or the individual farmer from providing ex situ provision of the material, because they would simply provide a duplicate to the gene bank, and then the gene bank would actually formally provide it to the user community. Have you considered this?
Actually, that is one of the best practices we have seen in the region here, especially like in the case of here in Uganda, where the gene bank actually goes to private individuals or communities and collects that material and keeps it in the gene bank, and also maps that area so that if the material is needed again in future, it can be accessed through the gene bank and not by directly going to the individuals to access that material. So this is for the case of Uganda. They have specifically said that if the material is to be used for food and agriculture, regardless of whoever is conserving it, you are to access it through the SMTA and through the National Gene Bank. So that reduces a lot of transaction costs related to negotiating and all these things, and also because these people don't know much. I mean, at the community level, the farmers really don't know what it means to give out your genetic resource and so on. So these transaction costs are already taken care of by the National Gene Bank people. And so this is also a very good thing that I think we can do in the SADC region, and especially here is where the SADC Plant Genetic Resources Center comes in as an institution that can back up this kind of system in the region, and also provide capacity to both providers and users on really how to have access to this material without a lot of these transaction costs. So that, I think that will be one of the propositions that we will discuss during the workshop. Thanks very much, Nigel.
Thank you for the question, Nigel. I'd just like to pass on to Prishni for any other question from the audience.
Yes, we have another question from Howard Mainbo. So it's addressed to Joanna. Taking into consideration the aspect of crop relative in respective protected areas, what are some of the in situ conservation approaches earmarked to be put in place?
Do you mean specific actions, conservation actions, or at the conservation planning level?
So I suppose within the protected areas for in situ conservation, yeah.
Well, I think, well,
if I may, Joanna, if you may, I don't know.
Okay.
I just give my 2 cents here. Yes, I think it's a very good question because it's really important to know what needs to be done. And we need to, once you have identified the important population within the conservation planning exercise, then measures need to be developed of how to conserve them in situ. In fact, we will be discussing that a little bit more tomorrow with the presentation from Jose. So there are certain guidelines that we've been developing under the European project of Farmers' Pride on how to manage wild populations of crop relatives. Joanna, if you want to add a few things, I don't know.
Yeah, I was just going to add, so crop wild relatives are wild plant species. So presumably the way you conserve crop wild relatives is the same way as you would conserve crop wild relatives. So the actions themselves can be various, and we will talk about them tomorrow, like Aishan said. But basically, these are wild plant species. So after knowing the populations, as Aishan said, and the status of the populations, we will have to act accordingly to conserve them.
Thank you, Joanna. Another question, Prishni, so we can maximize the number of questions from audience.
Yes, so from Sheila to Gloria, I suppose. So what do you anticipate will be the role of gene banks in access to in situ conserved germplasm, if at all?
I think there is the complementarity between in situ and ex situ conservation, especially of crop wild relatives, that we should not lose. So if some of these collections can be done, the crop wild relatives, and be kept in duplicates in the gene banks, it can make it easier even for their access, especially for breeders, such that they would only need to approach the gene bank to access them. But also, if the gene bank is involved in, let's say, mapping of these in situ materials and all this ecogeographic modeling, so that they have the knowledge of where these materials are, then anyone who wants to access them can easily just approach the gene bank. So the gene bank, in my opinion, will play a very, very central role, especially with respect to crop wild relatives that are in the list of 64 crops and in the public domain.
Thank you, Gloria. Another question, maybe we have time.
Well, in the same line, so Sheila asked that some of the access is directly from the protected areas. So who collects the material and ensures sufficient good quality seed?
Yes, actually, this is where the issues of institutional coordination comes in, and this is one of the gaps that we are already seeing in terms of protected area managers and gene banks working together, not only for in situ ex situ complementarity, but also for such simple things like collections, mapping, inventorizing of these crop wild relatives, and even providing access, because sometimes accessing the materials directly from protected areas has issues. If it is in the protected area and this protected area is under the Ministry of Environment, then you first have to approach the Ministry of Environment, and then the Ministry of Environment is the one who will determine which competent authority has to decide on the access of this material. And if they decide, okay, this material is being accessed through negotiations of pick and match and so on, it becomes difficult. And this is where the issue of transaction costs come in. So if there can be a clear process and a mechanism where institutions that both provide and use genetic materials can coordinate in terms of reducing the transactions related to accessing genetic material, then that would be the ideal situation.
Okay, thank you very much. The time is up now, 4 o'clock. Are there any other questions, Prishni, that we may have a minute or so
Well,
to respond?
some of the participants asked for the presentations if they will be available.
Yes, I think we can make those available, but I will need to discuss with the presenters first and get some PDF of those slides, and then we can share it with all those that have attended. Yes, we will try and organize that. Thank you. Well, I don't want to go over time, so I would like to thank you all for attending this webinar. This is just the first day. So tomorrow we will continue with two other very important presentations from Nigel Maxted and Jose Uriondo that I think will talk more to protected areas managers about actions on the ground, and also the experiences that we've been having on a sister project that we're implementing in Europe called Farmers Pride, experiences from that. So we look forward to rejoin us tomorrow at the same time tomorrow for the second day seminar. So thank you very much for today, and thank you very much to all the presenters and to Bioparma for their collaboration. Thank you very much. Goodbye.
Thank you. Bye.
Thank you. Bye-bye.
Thank you. Bye-bye.